Hello and welcome to Conception and Fetal Development. Today we will be going over conception and fertilization as well as fetal development. The first thing to keep in mind in regards to this is the difference between mitosis and meiosis, as both are important to conception and fetal development. Hopefully this information here is a refresher from your science classes. Mitosis occurs when a cellular division results in two diploid cells, each with 46 chromosomes. These cells are identical to the parent cell. In contrast, meiosis is unique to gametes and prepares the sperm and ova for fertilization. This type of division results in 4 cells each with a haploid number of chromosomes and occurs during 2 division times. The first division results in replicated chromosomes, after which the chromosomes intertwine and an exchange of genetic material happens and they then separate. The sharing of genetic information starts to form the various traits that will be present in the chromosomes. Following separation, the second meiotic division occurs and the 4 unique daughter cells are formed. At times, the chromosomes do not fully separate and there is extra genetic material that carries over to the final replication. Gametogenesis, or the formation of gametes for reproduction in both the males and females, begins with mitosis and ends with meiosis to prepare the sperm and ova. Oogenesis is the process by which the ova or female reproductive cell is formed. Prior to the birth of a female, a series of mitosis creates primary oocytes. At birth, a female is born with all of the primary oocytes she will ever have. After birth and throughout childhood, the primary oocytes lay dormant until puberty. Starting at puberty, with each reproductive cycle, one oocyte will undergo meiosis. The first phase of meiosis 1 occurs just prior to ovulation and results in the secondary oocyte and the first polar body. Meiosis 2 will only complete upon fertilization of the secondary oocyte to split into a second polar body and the ovum. The first polar body will also divide as seen in the picture. The final result is 3 polar bodies and 1 ovum. The polar bodies will disintegrate. Spermatogenesis is a lifelong process for males that begins in puberty and continues on. After puberty, there are continual mitotic divisions to produce primary spermatocytes. Eventually, each primary spermatocyte will undergo meiosis 1 to create 2 secondary spermatocytes. All secondary spermatocytes are with X or Y chromosomes. Meiosis 2 will then divide each secondary spermatocyte into 2 spermatids for a total of 4 spermatids from each primary spermatocyte. The spermatids then undergo changes to lose the cytoplasm and become sperm. Each ejaculation of the male contains an average of 200 to 500 million sperm, so it is clear that the spermatogenesis process is continually going at rapid rates to create the amount of sperm needed for successful reproduction. Fertilization is the formation of the zygote with 23 chromosomes from each parent. There are some key terms that must be known and understood to know the where and when of the fertilization process. The parts of the fallopian tube that are important are the ampulla and the fimbriae. The ampulla is the curved part of the fallopian tube and is the area where the sperm will likely meet the secondary oocyte. The fimbriae are the finger-like projections at the end of the fallopian tube close to the ovary that draw the oocyte into the fallopian tube, assisted by the motility under the influence of estrogen. The uterus has a few key portions that are also important. The fundus is the top part of the uterus and is the most muscular portion. At the bottom of the uterus is the cervix. The cervix is the opening to the uterus and has an internal and external os. The external os is the part of the opening that is at the very bottom, while the internal os is the top of the opening and the entrance to the uterus itself. Ova are fertile for 12 to 24 hours after ovulation, while sperm are fertile for about 2 to 3 days in the female reproductive tract. Some sources list sperm as fertile for about up to 5 days in very ideal conditions. This leaves a short window of opportunity for fertilization. The ovum has 2 layers of tissue that are pertinent to the fertilization process. The pink layer in the picture is the zona pellucida, and the elongated cells outside of that are the corona radiata. The ovum cannot move on its own and is instead moved along by the motility and contractility due to the estrogen influence in the reproductive tract. Remember that around the time of ovulation, the cervical mucus is thin and facilitates the movement of sperm through the cervix. Following ejaculation, the sperm propel themselves, using fructose for energy with the movement of their tails. Travel to the fallopian tube can take from 5 minutes to several hours. The movement in the fallopian tubes also helps to propel the sperm upward. During the travel, sperm undergo capacitation, or removal of the plasma membrane overlaying the acrosomal area, which prepares the sperm to be able to fertilize the ovum. This results in three things: the ability to undergo the acrosomal reaction, the ability to bind to the ovum, and hypermotility. The hypermotility further assists with the movement up the fallopian tube. Next is the acrosomal reaction, where acrosomes release enzymes to break down the corona radiata once the sperm has touched the zona pellucida. This allows the sperm to penetrate the zona pellucida. A block of polyspermy occurs at the moment of penetration of the zona pellucida and results in changes in the zona pellucida to prevent additional penetration by more sperm. Following penetration of the sperm, the secondary oocyte completes meiosis II. The nuclei of the two gametes approach and pair up to create a diploid number of chromosomes with unique genetic material. All characteristics are determined at this time, including the sex. Pre-embryonic development occurs during the first 14 days before implantation. There are many important changes that must occur prior to implantation. Following fertilization, the zygote is formed. There are many rapid divisions as the zygote moves down the fallopian tubes. These divisions are mitotic divisions and are called cleavage and results in cells known as blastomeres. These are all held together by the zona pellucida. The zygote is called a morolla once there are around 12 to 16 blastomeres. Once the morolla enters the uterus, there are some changes that occur. A central cavity forms inside the morolla, and this cavity contains a mass of cells called the blastocyst. The outer layer of cells replaces the zona pellucida to become the trophoblast. Multiplication is continued, and eventually the trophoblast will become the chorion, and the blastocyst will become the embryonic disc, which will develop into the embryo and the amnion. Early pregnancy factor is present and acts as an immunosuppressant. This is secreted by the trophoblast and allows for the foreign body, since the zygote has genetic material that is not maternal, to remain safe in the environment. Implantation starts with the attachment of the trophoblast to the endometrium for nourishment. This usually occurs in the upper posterior part of the uterus near the fundus. The zona pellucida then completely disappears around 7 to 10 days as the trophoblast has replaced it. This allows for the blastocyst to then burrow into the uterine lining until it is completely covered. The uterine lining, below the blastocyst, increases in depth and vascularity under the influence of progesterone. Following implantation, the endometrium becomes known as the decidua, with the decidua capsularis, which sounds like capsule, covering the blastocyst, while the decidua basalis is the part under the blastocyst and will develop into the maternal part of the placenta. The decidua vera lines the remainder of the uterus. Beyond cellular multiplication, there is a lot of cellular differentiation that must occur to form all of the various tissues and organs. The blastocyst differentiates into 3 layers around 10 to 14 days after conception. The ectoderm, mesoderm, and the endoderm, from which the embryonic disc and the amnion will be formed. The mesoderm and endoderm develop into all of the tissues, organs, and organ systems. The embryonic membranes are formed by the chorion, which developed from the trophoblast, and has chorionic villi, which are finger-like projections on the surface. The villi will eventually disintegrate except for the portion directly under the embryo, which become the fetal side of the placenta. The chorion is the outer layer of the embryonic membranes, while the amnion is the inner layer and is formed from the ectoderm. The amnion will contain the amniotic fluid and is where the fetus will develop. By the time of delivery, the chorion and amnion will have fused together. There are many functions of the amniotic fluid. Primary functions include thermoregulation, cushioning, allowing for symmetrical growth and no adherence of the fetus, and keeping the umbilical cord compression free. At 10 weeks, there is only around 30 milliliters of fluid, while this amount increases to around 700 to 1,000 after 20 weeks gestation and remains relatively consistent until the end of pregnancy, where it will decrease a little bit. Fluid allows for the fetus to practice respirations, and the fetus contributes to the amount over time by secreting urine. Oligohydramnios is a low level of amniotic fluid, while polyhydramnios or hydramnios is an excessive amount of fluid, usually over 2,000 milliliters. Given the knowledge that the fetus contributes to the amount by secreting urine, a low amount of fluid could be indicative of a renal problem in the fetus. The yolk sac is a second cavity in the blastocyst that's formed early in development and has the important function of forming primary red blood cells during the first 6 weeks. Following that time, the fetal liver takes over production of red blood cells. The yolk sac will eventually incorporate into the umbilical cord and disintegrate. The umbilical cord from the amnion contains one vein and two arteries in a normal development. Occasionally, there is one artery, which could be associated with other congenital malformations. The umbilical cord contains Wharton's jelly to prevent compression of the vessels. The cord has no sensory or motor innervation, so there's no pain when cutting the cord. The umbilical cord is normally inserted centrally on the placenta. Twins are often hard to understand and are frequently misunderstood by laypeople. However, this information is important for nurses to understand. There are two types of twins, fraternal and identical in lay terms. In medical terminology, fraternal twins are dizygotic and identical twins are monozygotic. You can see that each uses the term zygote, so in fraternal twins or dizygotic, the di indicates there are two zygotes, indicating there were two ovums that were fertilized separately, while in identical, the mono indicates that only one zygote was formed. In dizygotic twins, there are two placentas, two chorions, and two amnions, as each zygote develops separately into an embryo and eventually a fetus. There is a familial or genetic tendency to fraternal twins due to the tendency to produce more than one mature ovum with a reproductive cycle, so more than one ovum is released at ovulation. The genetic tendency lies within the mother's family. If the partner has twins in his family, it will not impact the genetic tendency, as the partner does not impact the mother's genetic makeup. In monozygotic twins, only one zygote is formed, and the zygote divides early in the pregnancy. All monozygotic twins will share a placenta. However, they may or may not share an amniotic sac. Division within the first 3 days after fertilization will create di-di twins. These twins each have their own amnion and chorion, so diamniotic, dichorionic, or di-di twins. This means they are in separate amniotic sacs. Division at around 5 days after conception results in monodi twins, or monochorionic diamniotic. And there is a single chorion, but two amnions, forming two separate amniotic sacs surrounded by a single chorion. Division after formation of the amnion results in mono-mono twins, so monochorionic and monoamniotic, and is very rare. These twins share an amniotic sac and are at risk for more complications due to the shared environment. The chance of having identical twins is completely random and does not have any genetic tendency. Development of the placenta starts with the chorionic villi as they form spaces in the deciduous basalis. These spaces will fill with maternal blood and the villi will grow into them. The villi will eventually differentiate into the cinctium and the cytotrophoblast. The cinctium is the outer layer and has direct contact with maternal blood in the inner villus spaces. The cytotrophoblast eventually thins and disappears around the 5th month, which allows for more exchange of substances. The cinctium is a placental layer that secretes hormones during the pregnancy. Some of the villi will become anchoring villi and will form various septa of the placenta. These subdivisions of the placenta are called cotyledons, and there are 15 to 20 total at term, apparent on the maternal side. Each cotyledon contains a vascular system allowing for utero-placental circulation and the exchange of gases and nutrients. Exchange of substances is limited in the first 5 months of pregnancy, but increases following the disappearance of the cytotrophoblast. Permeability decreases as the placenta ages, which is concerning if a pregnancy goes too far past the due date or if pregnancy conditions cause the placenta to age prematurely. The placenta weighs around 400 to 600 grams at birth. By 20 weeks gestation, the placenta covers half of the uterine surface and will only thicken after that point, but not get wider. Placental circulation is a key concept in regards to the placenta. The trophoblastic tissue on the fetal side begins to invade the decidual basalis, and the chorionic villi continue to grow, divide, and the fetal vessels form. Surrounding the areas where the fetal vessels and villi have reached towards the decidual basalis is the intervillous space. The intervillous space contains maternal blood that has pooled as the endometrial spiral arteries empty into this space. Exchange of gases occurs around the end of the 4th week, and the intervillous space is where the nutrients are exchanged. Waste products from the fetus diffuse into the intervillous space from the fetal vessels, and nutrients coming from the mother diffuse from the intervillous space into the fetal vessels. The umbilical vein is the source of transport of the fetal oxygenated blood to the fetus through the umbilical cord, while the two fetal arteries, or umbilical arteries, bring the unoxygenated blood back towards the placenta. This seems opposite of what we are used to, where the arteries carry the oxygenated blood and the veins carry the unoxygenated blood. The placenta has many functions, and these functions allow the placenta to achieve fetal respiration, nutrition, and excretion. Metabolic functions include the production of glycogens, cholesterol, and fatty acids, in addition to the storage of glycogen and iron. The placenta is able to break down histamine and epinephrine. Transport functions seem to be the most obvious functions of the placenta and can occur via simple diffusion, facilitated transport, or active transport. Those are those terms where that goes either against the wave or with it. These transport functions allow for diffusions of substances across the placenta. Oxygen, carbon dioxide, electrolytes, insulin, thyroid hormones, substances of abuse such as cocaine and nicotine, and many other substances are able to cross the placenta. Glucose crosses, but the fetus uses glucose quickly, so it must cross at a quick rate. Much of the glucose, while it can cross via simple diffusion, may need to be transported via active transport in order to accommodate the quick rate of glucose being utilized. Some viruses can cross the placenta in addition to some bacteria. Transportability is decreased with reduction in placental surface, as well as with edema, like an infection. Decreased blood flow, such as in hypertension, can also decrease exchange. The endocrine properties of the placenta produce HCG, or human chorionic gonadotropin. This supports the corpus luteum, and it also produces HPL, estrogen, and progesterone. By the 11th week, there's enough estrogen and progesterone produced by the placenta to support the pregnancy. The immunologic functions of the placenta allow for the placenta and the fetus to not be rejected. These are homografts, so generally they're rejected. This anti-rejection drugs similar to after transplant. Our bodies do not react this way to the placenta and embryo. We don't need anti-rejection drugs as we would after transplant. Progesterone and HCG also seem to help suppress this reaction along with the placenta. The fetal circulatory system is important to understand. There are a few things that make fetal circulation unique. The majority of the blood supply bypasses the fetal lungs, as the placenta assumes the role of providing oxygen and removing carbon dioxide during the pregnancy. The blood enters the fetus through the umbilical vein and approaches the liver. Only a small amount of blood will enter the liver, while most bypasses liver circulation through the fetal ductus venosus, to flow directly into the inferior vena cava. Blood then enters the right atrium, passes through the fetal foramen ovale into the left atrium. Blood then enters the left ventricle and is pumped into the aorta. Again, only a small amount of blood goes to the pulmonary artery to nourish the lungs. The blood does not need to go to the lungs for oxygenation, as it is already oxygenated. The majority of the blood enters the ductus arteriosus to then enter the descending aorta. After the blood has circulated, the unoxygenated blood returns through the fetal veins to the umbilical arteries to return to the placenta. In the developing fetus, blood with the highest oxygen is available to the vital tissues, while the lower levels of oxygen go to the non-vital tissues. We see a similar pattern after birth during times of significant illness. The fetal heart does have its own pacemaker. When the fetus feels stress, the sympathetic nervous system will cause a release of norepinephrine and increases the fetal heart rate. You will also see that during times of increased fetal activity, there is an increase in the fetal heart rate and variability. This is similar to what we see again after delivery as well. Lastly, we're going to talk about fetal development. While fetal development overall is quite complicated, there are a few key considerations to take note of throughout fetal development. The embryonic stage starts around day 15 and ends around the 8th week of pregnancy. During this time, the embryo reaches a crown to rump length of about 3 centimeters. This time is when the tissues are differentiating into essential organs and also when external features are developed. The embryonic stage is the most vulnerable stage for development. The embryo is very susceptible to teratogens, yet is the earliest part of pregnancy, and many may not even know they are pregnant at this early stage. Following the 8th week of pregnancy, the fetal stage is the remainder of the pregnancy, during which the body systems are perfected to prepare the fetus for life outside the womb. A full-term pregnancy is considered 40 weeks after the last menstrual period, or 38 weeks after conception. Full-term infants average 20 inches in length, and weight varies greatly. Amniotic fluid is around 500 milliliters as the fetus approaches term. During fetal development, there are a few key body systems that are often asked about by expecting parents. These are a great place to focus when looking at what develops when. For this presentation, I will discuss the cardiac, genitalia, gastrointestinal, respiratory, skin, and nervous system development. The fetal tubular heart begins to beat around 4 weeks after conception and can be detected by ultrasound around 7 weeks. The heartbeat can be heard by a Doppler around 8 to 12 weeks and by a fetoscope around 16 to 20 weeks. Around 8 weeks, the male and female genitalia will appear similar, but around 9 weeks, differentiation begins to occur. Determination of fetal sex can be done around 16 weeks. At term, the genitalia will be well developed in both sexes. However, preterm deliveries will have genitalia that is not yet fully developed. The fetus will begin to suck and swallow amniotic fluid around 20 weeks, and peristaltic movements will also begin. This allows the fetus to be able to prepare for eating and nourishing themselves outside the womb. Respiratory movements may occur between 24 and 40 weeks. The nostrils will reopen at 24 weeks, and the alveoli begin to appear in the lungs, allowing for the start of surfactant production and gas exchange to occur. This also is a key moment in indicating the age of viability of a fetus outside the womb. By 38 weeks, the lungs are generally mature enough to decrease the risk of ineffective surfactant production that could cause respiratory distress syndrome. Lanugo, or fine hair that covers the body, appears on the skin around 16 weeks and covers the body by 20 weeks. Brown fat and vernix begin to form around 20 weeks, and the lanugo then begins to disappear around 36 weeks. At term, vernix is present in skin creases, and there are creases covering the sole of the foot. At 24 weeks, the fetal brain looks like a mature brain, and at 28 weeks, the brain begins regulation of some body functions. Again, these are key components related to the age of viability and the ability for the fetus to survive outside the womb. The eyelids will open around 26 to 29 weeks, so infants born before that time will have fused eyelids. Thank you for listening today.